Hi, my name is Qingqing Wang, and I am a 2016 Arnold O. Beckman postdoctoral fellow. During my fellowship, I have guided my research to follow Dr. Beckman's philosophy, to generate and develop methods that enable the scientific community to make greater discoveries, and to be not afraid of taking risks. Before I became a fellow, I found that the gap between the computational and experimental biology would miss many opportunities to develop highly effective tools. The bioinformaticians would be unaware of the subtle aspects of the biology, and the experimentalists would be unaware of the potential changes that would otherwise greatly improve the analytical power of their work. So with my background in mathematics and the support from the foundation, I made the jump to gain hands-on training in modern biochemistry and genetics, and work at the intersection of large-scale computational analysis, software tool development, and modern experimental techniques. With this knowledge, I've developed analytical tools for fundamental biology and precision medicine. Now, let me give you a brief introduction.
All cells in our body inherit an essentially identical copy of DNA, yet this genetic information is decoded differentially in different cell types, resulting in diverse and distinct cell functions and behaviors. One major contribution to this biological diversity is that multiple and functionally distinct mRNA and protein isoforms can be generated from the same gene locus. So one big question I'd like to ask in my research is how is this isoform complexly modulated to specify a cell's identity and behavior, and how misregulation of these isoforms can lead to diverse pathological consequences. But before I can ask these questions, I need to be first able to identify mRNA isoform profiles from different samples. This is not a trivial question because RNA profiles can be extremely complex. They differ from cell type to cell type and also from different developmental stages. For this purpose, I developed a software tool called JOM for deciphering the incredible complexity of RNA profiles. Starting from RNA sequencing datasets, JOM can provide a quantitative RNA isoform atlas. It will tell you how many total alternative usage of RNA isoforms are there in the sample, and also what patterns are these isoforms, as well as the quantitative relative levels of each isoform. Using JOM, I'm now able to investigate a large variety of cellular processes and investigate how RNA regulation is playing a role in it. As one application, I investigated mRNA isoform regulation in circadian rhythm control. This is an important process closely related to a human's daily routine, insomnia, as well as jet lag. There are different subsets of neurons in charge of different aspects of the circadian rhythm. Some cells regulate sleep, some cells regulate evening activities, while some cells regulate waking up. I looked at each cell population's isoform diversity as well as tracked their changes in the isoform profiles throughout the day, from sunrise to sunset. Strikingly, I found that each subpopulation of neurons possess their own very distinct mRNA isoform profiles that is closely related to each of their functions. I also discovered a subset of RNA isoforms whose alternative usage cycles throughout the day. These isoforms are in genes regulating neuron-to-neuron connectivity. In summary, I invented the method called JOM that catalogs and profiles signature mRNA isoform profiles in diverse cell types, cellular states, as well as individual patient disease samples. This method now allows us to investigate regulatory mechanisms that specify distinct mRNA isoform profiles corresponding to each cell state. It also allows us to investigate the phenotypic and pathological outcomes from different usages of these mRNA isoform profiles. Now, for my pathological applications, I've been focusing on cancer and neurodegeneration diseases. I'd like to thank my collaborators both inside and outside of UC Berkeley during my fellowship terms. Specifically, I'd like to thank the Arnold O. Beckman Foundation for their tremendous support to my research, as well as for building such an outstanding community, which I am proud to be a part of.